So now the next application of this alpha particle lies in a great therapy, which we call as the boron neutron capture therapy. So before we go deeper inside these therapies, so let us have a brief idea that for boron neutron capture therapy, we need a nuclide, which is boron 10. And one of the most important property of boron 10 is that it has a higher cross section for interaction with thermal neutrons. So neutrons can interact with boron 10 very effectively because it has a higher cross section. And one more thing is that this boron 10 is taken mostly by the neoplastic tissues or neoplastic cell. And why neoplastic cells takes boron 10, we'll talk about it later. But as of now, you should keep in mind that boron 10 is taken more by the neoplastic tissue. And the absorption of neutron, it converts boron to lithium and it emits an alpha particle. And we know that alpha particles has high reality, it deposits energy over short range, and it kills the cell by creating molecular lesions like double strand breaks, single strand breaks, or unrepaired or higher number of unrepaired double strand breaks, and thus it kills the cell. So now we can see over here that these boron 10 are preferentially taken by the neoplastic cells. In this case, you can see that there is only one, but there are many boron 10s taken by the neoplastic cell. So there are many higher number of boron 10 in these cancerous cell. And when we irradiate these cancerous cell with thermal neutron, this boron 10 captures these neutron and converts to lithium 7 and emits an alpha particle. And this alpha particle then goes and attacks the DNA, causes double-strand DNA breaks and multiple molecular lesions and unrepairable double-strand breaks, which in turn kills the cell. And now that's the main part, that how specific this therapy is, because boron 10, as I've told before, it's taken in excessive amounts by the neoplastic cell, and normal cell does not take boron 10 or takes basal uptake of boron 10. So as this cancerous cell takes more boron 10, so the probability that this neutron will interact with these cancerous with these cells rises, and thus this N alpha reaction increases in the cancerous cell, and we have more amount of alpha particles released in this cancerous cell. But whereas normal cell do not have enough boron 10, so in the interaction of these neutron to these cells are also poor, so this therapy is not effective. So alpha particles which are emitted in this region are very less, but we have more amount of alpha particles in these regions, and thus it is now depositing energy over these neoplastic regions. So that's what we want from therapy, that we have to deposit more energy to the cancerous cell, and we are sparing the normal cell. So that's a basic idea regarding BNCT. So that's a small animation which I will play for you guys. So that's a particle accelerator. So what it does, it accelerates proton. And here we have a beryllium source. So what basically happens that when you bombard proton to beryllium, it results in a daughter nuclide, let it be X, and emits neutron. And over here we are getting beam of neutrons, fine. But these beam of neutrons are so high in energy, and that's why we have to do something over here. So we have to reduce the speed of neutrons, and there are different mechanisms of reducing the speed of neutron. And that's how, at the outcome of this, and because of this mechanism over here, we are getting neutrons which are thermalized. Fine, they don't have higher velocity. So if they have higher velocity, then the probability that this neutron will interact with this boron 10 is reduced. And that's why we have to reduce the velocity to make this neutron thermal, so that this reaction becomes much more effective and lead to higher production of alpha particles in the region and higher cell death. So now we see what goes over here. So that's so this BNCT is very important for treating inoperable cancer, also for invasive cancer, or for those cancer which does not respond to the traditional radiation therapy. So what we do next basically is that that's the solution that we infuse, and that's a special solution because it contains boron 10 along with a amino acid. And that's what we call as boronophenylalanin. So that's boronophenylalanin, and we know amino acid. This phenylalanine is a very important amino acid and essential amino acid. So this boronophenylalanin is a combination of boron 10 and the amino acid phenylalanine. And we know that the tumor cells are highly glycolytic in nature. They express higher amount of glucose transporting proteins on the cell surface. And apart from these tumor cells also express higher amount of amino acid transporting proteins too. Fine, as we can see over here that as because our the cancerous cell has higher expression of these amino acid transporting proteins, and with these transporting proteins, this boronophenylalanin enters entry inside the cell. And this cancerous cell take more amount of boronophenylalanin than normal cell, and thus we can see there is higher amount of boronophenylalanin inside the cancerous cell, whereas there are very minimal amount of boronophenylalanin in normal cell. And thus when we irradiate this tumor with a beam of thermal neutrons, so now what we are doing, we are irradiating this tumor with a beam of thermal neutron. Then the reaction happens, then boron captures the neutron and results in production of lithium-7 and alpha particle. And those alpha particle goes and attacks the DNA of the cell, and it leads to the shrinkage, necrosis of the cell, and hence we can see how beautiful this therapy works. And we can see that we are irradiating this cell. So the probability of these interactions are higher in this cell because it has higher fractions or higher numbers of this boron 10, whereas normal cell has very poor number of boron 10. So the probability of interaction is very less or negligible in normal cell. So what we are doing, we are giving a localized therapy, a very precise therapy to the cancerous cell, and leading to effective therapy response. So that's example of the alpha particles in medical sciences. So that's boron neutron capture therapy. So now, after discussing about alpha decay process, we'll now move forward to beta decay process. So what is beta decay? So this beta